Welcome to MVIF Conversations, the podcast.
Hi, everyone, and welcome to the MVIF podcast series. I am Pamela Ferretti, a postdoc at the University of Chicago, where I study early life microbiome in human and animal populations. I am very excited today to be hosting today's episode of MVIF. Today, we're diving into a part of the microbial world that is still largely unexplored, but full of surprises, the microbiome of wild animals. When people hear animal microbiome, they often think about pets and livestock, but there is a whole world out there, from wolves to giraffes to birds, and we know surprisingly little about the microbes that live inside these animals and how those microbes shape their health, survival, and evolution. To explore the world of wild animal microbiome, I'm joined by Elin Videvall, researcher at the Uppsala University in Sweden. Dr. Videvall focuses on studying the microbial communities that live inside wild animals and how these microbes interact with their hosts and impact their health. Using DNA sequencing and computational tools, her lab studies how factors like diet, environment, and the animal's own biology shape the composition and function of their microbiome. More broadly, her research connects evolution, ecology, and conservation, helping us understand how microbes influence animal health and survival in the wild. Elin, welcome to the podcast.
Thank you so much for having me. I'm very excited to be here.
So I would love to start from the beginning. Studying the microbiome of wild animals is quite a unique path. How did you get into this field?
Well, I've always been interested in wild animals. I think even as a child, like growing up, I loved watching nature documentaries and I was reading popular science books. And then I chose to study ecology and evolutionary biology at university. And I did my PhD on microbes in birds, mostly pathogenic ones like avian malaria. But then one of my projects was about ostriches in South Africa, and it was to investigate this very high mortality they had because of supposedly enteritis, which is a gut disease. And back in the day, people were very focused on finding sort of the single pathogenic culprit that was causing a disease. But as time went on, I started thinking more and more about actually maybe they are missing important microbes, these ostriches, especially because at ostrich farms, they were required to hatch and raise chicks separately from adults because of regulations they have regarding avian flu. So this is the time point when I started studying microbiomes.
That's incredibly fascinating. So you mentioned your work on ostrich microbiome and survival in early life. What were your findings exactly?
Yes, so one of the things I wanted to do is an experiment where instead of giving the ostrich chicks antibiotics, instead, I gave them feces from the adults, like literally served on a platter to them, because I still wanted to make it voluntary for them. But they absolutely loved eating that. They were happily munching on that. And then when I analyzed the data afterwards, I could see that these chicks that had access to adult microbes, they did so much better than the controls. So they grew faster, they had lower mortality, and they acquired a microbiome that was sort of more mature and more diverse, which I thought was really fascinating. So I definitely felt like gut microbiome is something that is extremely important for animals and wild animals too. But at the time, the literature was almost non-existent. There were so few studies, and most of what we did know was from the human microbiome field.
Yeah. So I find your experiment that you just described particularly interesting because it really shows that it's possible to modify the microbiome of these ostriches by simply exposing them to the microbes of their parents, of adult individuals, and that this can actually improve the chick's survival and growth. And now, if I understand correctly, you are kind of taking this question into a different system. You're now working on reindeer cows, trying to understand whether the microbiome plays a role in their growth and survival as well. Is that correct?
Yeah, exactly. So one of our projects in our lab is about reindeer calves and how their early life gut microbiome is related to their health. So we're looking into things like mortality, the causes of mortality, because they have different causes, how they grow over the summer, parasite diversity, parasite load, and also how the gut microbiota is shaped by what they eat. And in that regard, I think reindeer, it's a very interesting animal to study from a microbiota perspective because they are ruminants like cows. And that means that they are critically dependent on the microbes to break down and ferment the food they eat. But they are not domesticated. So I usually refer to them as semi-domesticated. So in Sweden, the reindeer, they are owned by the indigenous Sami reindeer communities. So they gather them twice a year to mark them. And that's really valuable from a scientist perspective because it gives us the opportunity to sample them and weigh the calves and all that. But in all other aspects, they're much more like wild animals because they roam freely across these vast natural landscapes. They eat things like lichens, mushroom, birch. They mate freely, and they're also heavily subjected to natural selection, such as predators, parasites, diseases, weather, and so on. So all of these things, it makes me think that their microbiomes are very natural in that sense, more like wild animals than captive animals.
Yeah. So here the idea is not just to describe the microbiome, but really to understand whether early life microbiome can predict things like growth, health, and even mortality. And I think this is particularly exciting because in wild populations, in many cases, the cause of death remains unknown. So the microbiome might well be one of the missing pieces. So if we zoom out a little bit, this idea of the microbiome affecting survival also connects to a much broader question, which is conservation. Because the microbiome might not only matter for the health of individual animals, but also for the success of conservation efforts for entire species. So, for example, when animals are bred in captivity to be then released into the wild, could you tell us more about how the microbiome might play a role in these animals?
Yes, so I've been interested in this question about how captivity changes animals' microbiome for some time now. And there's been quite a few studies, and most do find quite striking differences in the microbiome of wild animals versus the same species in captivity. And it's tricky to pinpoint the causes because I think there are several causes playing a role here. So diet is of course very different in captivity, but not only diet. In captivity, animals also get medications, they get vaccines, veterinary care, and they live in these human-built enclosures that can be quite sterile, especially compared to nature. And also there have been some neat studies showing that they acquire some microbes from the human keepers as well. So this question of captivity is it's of course important for the animals that live in captivity today. But another reason why I think it's important to study is for conservation purposes. So in instances where we do things like captive breeding of species, which we later want to release into the wild, if the microbiomes of captive animals are so different compared to the wild counterparts, what happens when we release them? Will they be less adapted to digest their new food in the wild, or will they be less adapted to resist things like diseases and parasites? We really don't know the answer to these questions. So we've done some pilot studies in horses and bison of this concept called fertilization. So fertilization is kind of the opposite of domestication, where we've been comparing feral wild individuals with domesticated or captive ones. And we always find that the gut microbiomes of feral populations are very different. And to give you some sort of example of how important this can be, this type of research. So one thing that comes to mind is this conservation project where they raised black grouse in captivity for release. So grouse is a type of bird that eats these very fibrous, hard to digest food items like birch, willow buds. But in captivity, they were given this commercial chicken feed. And so what happens is that I suspect that their gut microbiota wasn't properly adapted to the new environment, because when they released, they weren't able to digest the food properly, and they became very malnourished. And then most of the released individuals died from starvation, or they were taken by predators.
This example is incredibly striking. So animals that are perfectly healthy in captivity might actually fail to survive in the wild, potentially because their microbiome is not sufficiently adapted to digesting their natural diet or just the surrounding environment in which they are released into. And this raises a very important point. When we think about conservation, we usually think about genetics, habitat, and population size, but we rarely think about microbes. And yet they might be essential for animals to actually survive and being able to reproduce in their natural environment. Another aspect of your work, Elin, that I find really fascinating is that not all animals follow the same rules. In humans and many mammals, we often think of the microbiome as relatively stable and shaped by factors like diet and other host factors. But some animals seem to break these rules. You mentioned birds, for example.
Yeah, so one aspect I think is so fascinating with about wild animal microbiomes is that, and I talk about this often with people, that birds are so very different from other animals, especially mammals. And if you look at mammals and birds objectively, they should be very similar in their microbiome composition. Both of these groups of animals, they are endotherms, they raise their young, they occupy similar food niches. Some eat fruit and some eat fish and so on. But now we know, thanks to some really cool studies in the beginning from the Night Lab and from other people, that bird microbiomes are really quite different. One thing is that mammals typically have a signal, for a host phylogenetic signal in the microbiomes, which is called phylosymbiosis. But birds do not have that. So that means that there are different ecological and evolutionary processes sort of underlying how these microbiomes are shaped in these two animal groups. And thanks to several studies of wild birds, we now know that they acquire a lot of microbes from their local environment. So they often show this geographic structure, and bird microbiomes often varies a lot, both between and within individuals.
And this is really interesting because it challenges our assumptions. If there are some animals that do not have a stable microbiome, or if the microbiome is mostly shaped by their surrounding environment, then we need to think again, to rethink some of the concepts we take for granted from human studies. If I'm also thinking, for example, I'm also thinking about bats, which show similar patterns. And studying these outlier systems might actually help us understand more general principles of host-microbe interactions.
Yes, so bats are cool. They're kind of like birds in that sense. And because of emerging results like these, I think it's really important for us that we do not simply assume that animal microbiomes function or are governed in the same way that human microbiomes are. So, for example, we found that in wild giraffes, we found that the diet composition and the diet diversity is not linked with their gut microbiome. Instead, there seem to be other factors that are playing a role. And as I mentioned, with the birds too, their microbiome tend to behave very, very differently from humans.
That's very fascinating. So let's talk about the practical side. Studying wild animals sounds incredibly exciting, but also extremely challenging. What are the biggest obstacles that you face when studying these animals?
Yes, so the first one that comes to my mind is, of course, funding. I think it's difficult enough to get funded to study wildlife, but even harder to convince review panels that we actually need to study microbes inside wildlife because so little has been done so far. So it's tricky to sort of point a specific study to say, these are the things that are so cool. And the second challenge, I would say, is definitely related to sampling, sort of finding fresh fecal samples or waiting for wild animals to defecate. And most wild animals are not ID tagged visually. So when we do sample fecal samples, sometimes we're not able to link the samples with a specific individual. And this is a problem that we had with the feral horses, for example, because they all look identical. And so we're not able to tell them apart, and we're not able to mark them either. So that can be tricky. But we solved this in a larger project that we have on wolverines, because in this project, we're lucky to collaborate with people who genotype the fecal samples using a SNP chip. So in this project, we have lots of metadata on each individual, which is really neat. And in another large project we have on flycatchers, where we study diet and microbiomes. So flycatchers are these small insectivorous birds. And because they breed in nest boxes, we are able to catch all of them already as nestlings, and we can give them these ID rings around their legs when we sample them. So that's really valuable.
Nice. So from what you told me, I gathered that collecting samples and metadata seems to be important bottlenecks in your research, given that you cannot follow these animals in the wilderness all the time. Once you have managed to overcome these obstacles related to sampling, do you also face other difficulties when analyzing their data?
Yes, definitely. The one thing that comes to my mind is the microbial databases. They skew quite heavily toward human and model organisms. So when we study the microbes of wild animals, there will be lots more unknown microbes. And I can give you an example of this. Some time ago, I sequenced the giraffe's gut microbiome using a method called deep pack bio shotgun metagenomics to look at the bacterial genomes. And it's partly because we were curious to what we would find. And after we had assembled all the bacterial genomes, which had there were a lot more of them than it were in humans, so it was very diverse, we could tell that 98.5% of all the bacteria, they were species that were completely unknown to science. So we didn't know what species they were. And that's still, a giraffe is a ruminant, so I thought maybe there would be some known ruminant microbes in there, but there's so many unknown microbes. And to me, that sort of demonstrates just how little we know about this vast hidden biodiversity of microbes living together with wild animals.
I agree. This has been a really fascinating discussion, Elin. What really stands out to me is how much we still do not know, and also how much potential there is in studying these systems. From early life survival to conservation and even broader biological insights, really wild animal microbiomes kind of open up a completely new perspective and a completely new, still very much unexplored world. So Elin, thank you so much for joining me today.
Thank you, Pamela. Thank you so much for inviting me. I really enjoyed this discussion. And if I can just take a minute, I want to mention that I will be going to the ISME conference in New Zealand in August. So I'm organizing a session there on microbiomes in conservation. So I'm happy to chat about microbiomes or wildlife microbiomes or microbiomes in conservation with anyone who are going there. It will be very neat to see people interested in this topic.
That's wonderful. Thank you so much, Elin. And thank you for everyone listening. If you have enjoyed this episode, stay tuned for more conversations on microbiomes across different systems. See you next time.
Thank you for listening to MVIF Conversations, the podcast. Until next time.